Hi, my name is Kirili Stauch, and I'll be presenting on research on a comparison of vapor and feeding ethanol exposure in honeybees, Apis mellifera. The main purpose of the study was to do a couple of things. So first, I wanted to do a comparison of the honeybee hemolymph levels, looking at the traditional drinking method compared to the new vapor method. Now, the drinking method is usually used with the honeybees, while the vapor method has been used mainly with the fruit flies. And I wanted to create a dose-response curve for the vapor method and compare that to the traditional feeding method dose-response curve, as well as to collect baseline data so that we would have an idea of how much is actually being metabolized by the honeybee with the feeding method in comparison to the vapor method. Now, there have been 5 studies in the past that have used the vapor system with the honeybee. However, these did not establish dose-response curves, and they also didn't know the exact dosages used for the studies with the vapor method. And so it's very important to build this background information to use this method in future research. The honeybee model is an ideal alcohol model for a couple of different reasons. First, the honeybee genome has been heavily sequenced. This allows researchers to look at genetic impacts of alcohol, as well as to look at different mechanisms behind alcohol. Now, they're complex social creatures, and this allows researchers to study different impacts of alcohol on learning, behavior, social behavior, and biological mechanisms. And the unique environment of the honeybee and their caste systems allows researchers to really look at these complex behaviors. Additionally, there are a lot of similarities to human behaviors following alcohol consumption, and this is something that's seen throughout the honeybee literature, where you're seeing similar behavior effects, as well as similar effects on other systems with the honeybee, such as the hemolymph levels. For this experiment, I used 316 honeybees, and these were recruited from the Oklahoma State Comparative Psychology and Behavioral Biology hives. The bees were collected using falcon tubes off of a feeder. This method was used to ensure that the honeybees were actually forager bees and is used as an experimental control, so that way all the bees are around the same age. Once bees were collected, then they're brought back to the laboratory where they're exposed to alcohol using one of 2 methods. Bees that were exposed following the feeding method were harnessed into bullet casings and then fed 10 microliters of sucrose and ethanol solution based on their concentration group. And these solutions were either 0%, 1%, 2.5%, 5%, or 10% ethanol. Now, bees in the vapor exposure groups were placed in a tube, as you can see in this image here, and then a timer was set based on their exposure time. And their exposure times were either 0, 1 minute, 2.5 minutes, 5 minutes, 10 minutes, or 20 minutes. And as you can see here, there were ways to manage the alcohol flow so that the alcohol flow maintained the same amount of flow for each honeybee. Then for both groups, once the timers were done, then the timers were set based on blood draw time, and they were placed to the side until it was time for their blood draw. In addition to the ethanol hemolymph that was collected later on, which I'll be talking about in the next slide, we also looked at the behavior of the vapor bees specifically. The reason we did this was because the vapor bees were not harnessed, while the harnessed bees, we would have had to unharness them in order to look at their behavior. And due to this, we only observed the vapor bees. And then we recorded, while they're being exposed, the time that they lost balance. Following the blood draw timer going off, the bee was placed in the freezer. Then once the bee was fully sedated, a pair of her wings were amputated. And then a 1 microliter microcapillary tube was placed at the site of the wound. Then she was pricked with a scalpel if the hemolymph wasn't flowing. And then that was combined with 9 microliters of a butanol solution for storage. And then the sample was placed in the freezer until it was run using PCR analysis. All data analysis was conducted in SPSS statistics. For the loss of balance data, a logistic regression was conducted. And this logistic regression was looking at alcohol exposure and blood draw time, with the loss of balance data correctly predicted for 82.9% of the total cases. It also showed that loss of balance was observed in the higher alcohol concentrations compared to lower alcohol concentrations. And this is as expected with the literature. A lot of the literature shows that the honeybees lose balance or have motor function impairments when it comes to higher alcohol concentrations compared to lower alcohol concentrations. Additionally, blood draw time did not increase the odds of loss of balance. A pair of linear regressions was conducted to look at the ethanol hemolymph levels and the effects of the factors of ethanol exposure and blood draw time on these levels. Ethanol exposure and blood draw times significantly affected ethanol hemolymph levels in the vapor bees, but not in the feeding bees. For the vapor bees, ethanol exposure and ethanol hemolymph levels were significantly correlated, while blood draw time and vapor bee ethanol hemolymph levels were not. In the feeding bees, like the vapor bees, the ethanol dosage and ethanol hemolymph levels were significantly correlated, while the ethanol dosage and blood draw time with ethanol hemolymph levels were not. The figure on the left here shows the loss of balance data, and as you can see on the graph, that only the bees in the 5%, 10%, and 20% groups for the vapor alcohol bees lost balance. And as you can also see that the loss of balance percentage increased linearly from 2.5 through 10-minute exposure, but then the increase slowed once it reached over that 10 minutes. Now on the right side, you can see a graph of the Oklahoma vapor and feeding bees with the feeding bees in the 2A, while the vapor bees in the 2B. And so as you can see here, the vapor bees have a significantly higher ethanol hemolymph levels, especially when you see the higher levels of ethanol exposure or dosage. We learned a couple of things from this experiment. First, we learned that we were able to compare the vapor and the feeding method, and we were able to find that the vapor bees had higher ethanol hemolymph levels overall. And this is probably due to the way the vapor ethanol is metabolized compared to the feeding ethanol that's orally consumed. Now, when it's being processed through the vapor inhalation method, the ethanol is metabolized a lot quicker because it bypasses the digestive system, so it goes into the bloodstream quicker. We do believe that the equivalent dosing is possible via the feeding method and the vapor method, but only for ethanol hemolymph levels that are less than 40 millimolars. So this means that for the higher levels, that we would have to adjust the dosing in order to determine how much of the ethanol for the vapor system is equivalent to these higher levels of ethanol dosage. For future research, we suggest that researchers look into alcohol and genetics and look at some of the genetic factors behind this metabolization of alcohol, but also looking at the impacts that alcohol has on genetics